Good afternoon, ladies and gentlemen. My name is Torsten Hombeck, Chief Commercial and Strategy Officer of Promethera. I'm going to spend the next 15 minutes, hopefully not more than 15, with you, and I know it's the second last presentation of the day, to talk about Promethera, quick overview of who we are, and then also to talk about our clinical stage development pipeline. Our vision is to be the leader in the cell therapy space with innovative technologies originated from the human liver. And we want to do this by continuing developing our clinical stage cell therapies to address a large spectrum of liver diseases with no effective therapeutic cure. We have recently acquired the majority of the assets of another key player in this field, namely Cytonet, a German cell therapy company focused on liver diseases. And together with the assets of Cytonet, we are now joining complementary facilities and innovative cell therapies to treat both inborn genetic diseases but also acquired liver diseases. Here in the US, we have operations in Durham in North Carolina, with GMP-compliant manufacturing facilities. And with that, we also have access to the US organ procurement organizations from which we get our human livers from in the US. In Belgium, about half hour south of Brussels, we have our headquarters with GMP-compliant manufacturing facilities and also our R&D labs. So our clinical development program looks as such. We have two cell lines in clinical development. One is HepResc, and I'm going to talk to you about this in a second. And the other one, which is the core cell line for Promethera from a strategic perspective, which is HepStem. HepResc has been clinically evaluated in two clinical trials in urea cycle disorder patients. And we are in the preparations of preparing a new drug submission with Health Canada, and we target to get that paperwork in with Health Canada by the beginning of next year. We have HepStem in one clinical development program and are going to initiate another one very, very soon. The program that is currently enrolling patients also enrolls patients in urea cycle disorders. And we plan to start a phase 2 clinical stage trial in acute and chronic liver failure patients. Two other programs which are currently in preclinical stage in animal models is HepStem in NASH and HepStem in fibrosis. And based on hopefully positive animal model data, we would like to initiate clinical trials in these indications as well. Let's start talking about HepResc in urea cycle disorders. From a technology or manufacturing perspective, HepResc is an off-the-shelf allogeneic product. It exists of mature hepatocytes, and we get the human livers that are unsuitable for transplantation from the OPO network here in the US. And from a manufacturing perspective, the liver is enzymatically digested into a purified hepatocyte suspension, which is cryopreserved at a temperature of minus 130 degrees centigrade. And at the time of administration at the hospital, the drug product is reconstituted with its diluent and then infused into the patient. The target population for HepResc in UCD are very young patients, very early onset UCD patients with severe deficiencies. These patients cannot tolerate the protein from their nutrition and build up very dangerous, risky levels of ammonia in their blood, which often lead to either death in severe cases or severe irreversible brain damage. And the only cure that is available that helps these patients is a liver transplantation. Now, a liver transplantation at week 1 or month 1 of these patients' lives is impossible and very, very high risky. And what you need to do is to bridge these patients from the time of diagnosis to the time when the patients are a little bit older, a little bit bigger, also from a size and weight perspective, and can then be transplanted. And that is, in fact, the treatment paradigm or the treatment suggestion for HEPA-RESC, namely a bridge to transplant from time to diagnosis to time to transplantation. And what we do is we infuse the purified human hepatocytes, mature hepatocytes, in the liver with the help of a catheter through the portal vein. These cells end up in the liver, engraft in the liver. The engraftment process takes about 2 to 4 weeks. And then these cells, these mature hepatocytes, take over the missing function, enzyme function, of the otherwise structurally functional liver in these patients. We have conducted 2 clinical trials. CCD02, upper left-hand side, was a phase 2 safety and efficacy study in Europe, and CCD05 was a phase C safety and efficacy study in North America, including Canada. We've enrolled 22 patients in the study, in these 2 studies, and we have this is, I think, a very nice slide that shows some efficacy data. We have been able to show that patients who have received HEPA-RESC are doing a lot better in terms of building ammonia levels or reducing ammonia crisis after the liver cell infusion compared to before the liver cell infusion. So what you see here on the slide are levels of ammonia to the left-hand side of day 0. You see it on the bottom, or the LCT on the top of that chart is the time when the liver infusion took place. And you see very dangerous levels of ammonia in the blood of these patients on the left-hand side prior to the administration of HEPA-RESC, and you see over a period of almost 360 days, namely 1 year, metabolic stability of these patients with no severe, maybe not even moderate crises. So these babies were able to be stabilized in their environment. They were able to tolerate protein so that they can grow and can ultimately be listed and received a liver transplantation to then have a normal life. So that was the program that was around our HEPA-RESC, our mature hepatocyte cell line. I now want to talk about HEPA-STEM and the development that we have done so far in this first indication, urea cycle disorders. HEPA-STEM, from a technology perspective, is also an off-the-shelf allogeneic product. We get the human livers from healthy disease donors via the tissue bank in Belgium at Saint-Luc. When this liver is, or these livers are enzymatically digested into a cell suspension. And we then culture this suspension, and during that cell expansion process, the mature hepatocytes die, and what emerge are the progenitor cells that then further multiply in this process. After these progenitor cells are harvested, they get cryopreserved at minus 130 degrees centigrade. Until they are administered in the hospital as a drug product. They are reconstituted with its diluent and then infused as a final drug product into the patients. So with HEPA-STEM in UCD, we target these kind of patients that are more the mild and moderate cases. And what we want to do is we want to improve the phenotype and their quality of life. So we target patients or children with a poor quality of life with urea cycle disorders. The effect that we have seen with HEPA-RESC was a very short-term effect. The engraftment took place relatively quickly, and then once these cells engrafted, were able to provide the missing enzyme. The HEPA-STEM effect is a longer-term efficacy effect. We have conducted several clinical trials. One is HEP-001, which was a phase 1/2 primarily safety study with some early readouts for preliminary efficacy. This study completed in 2014. We are currently enrolling patients into HEP002. It's an ongoing phase 2 study to further evaluate efficacy and safety in the same target population. Patients from HEP001 were enrolled into our safety SAF001 ongoing surveillance study, where we have a 48-month period over which we survey HEPA-STEM for safety. And then we have an observational study going, OPS001, which is a non-interventional retrospective and prospective observational study to better understand the disease. HEP001 was a study that was an open-label dose escalation study with 3 different dose levels of 1 cycle of HEPA-STEM designed to evaluate, as I said, safety and explore preliminary efficacy in patients in pediatric patients with either UCD, urea cycle disorders, or another inherited liver metabolic disorder, namely Crigler-Najjar. We have enrolled 20 patients, of which 14 had UCD and 6 patients had Crigler-Najjar. There was 1 infusion over 1 to 4 days, followed by a 30-day, 6 months, and a 12 months then follow-up. And based on that study that completed, we have designed a phase 2 study, HEP002, which is a prospective open-label multi-country efficacy and safety study of several infusions of HEPA-STEM in urea cycle disorder patients below 12 years of age. So that's the inclusion criteria, less than 12 years of age. And we want to, as a primary objective, get the functional efficacy after 6 months post first infusion, and evaluate safety as a secondary objective, and evaluate other functional clinical parameters in this patient population. The study is split into 4 phases. After the initial screening phase, there is a baseline phase where we have a 13C test and test the urea capacity of these patients prior to treatment. We then have a treatment period over approximately 90 days with several infusions of HEPA-STEM. And then we have a follow-up period of then another 9 months, so a total of approximately 12 months for efficacy evaluation. The target, we want to enroll 20 patients in this study. But what we do is we have an interim risk-benefit assessment of 5 evaluable patients after the 6 months follow-up period. And based on the risk-benefit assessment of these first 5 patients, we then decide whether to continue and enroll an additional 15 or stop the study. And the last couple slides, I want to talk about the same cell line, also HEPA-STEM, and now in the development of acute inflammatory liver diseases. HEPA-STEM in general has multiple potentials, and we see 3 therapeutic pillars for HEPA-STEM. One is a immunomodulatory effect, which can control the inflammatory processes. It's a regenerative. It has a regenerative potential to be able to repair tissue, and we see a potential of protein expression to replace either missing enzymes or to secrete other missing factors. So in acute and chronic liver failure, you see a typical progression of the disease as such. Start from the top left. You have a chronic underlying disease that often is triggered by maybe alcohol or other factors such as hepatitis that causes an inflammation in the liver. And that progression of the disease can lead to first fibrosis and ultimately cirrhosis of the liver. And if you have a second hit or acute insult in that process with an underlying chronic liver failure, you talk about the acute and chronic liver failure indication. Theoretically, it's a reversible event because the liver is an amazing organ and can regenerate itself. However, the short-term mortality in that patient population is about 50% in severe cases. And the only cure, the only thing to do to help these patients if they are that far in the if they have that far progressed in this disease is a liver transplantation. So we have designed a HEP 101 study. It's a phase 1/2 safety study where we want to enroll 12 patients with acute and chronic liver failure. We want to evaluate 2 dose regimens for up to 28 days as a primary objective, with a 3 months and 1 year post first infusion follow-up, and a follow-up for 1 year also with regards to safety. The inclusion criteria is grade 1 and 2 patients where we see a mortality rate of 20 to 30%. And this is a study that is about to start. In fact, we're looking forward to initiate not only the sites, but then to also report that the first patient has been enrolled. So as a summary, with HEPARESC and HEPASTEM, we have 2 new complementary cell therapies in clinical development for UCD, addressing distinct segments in this indication. And with HEPASTEM, we will soon evaluate in a clinical study the immunomodulatory effect of HEPASTEM in inflammatory liver disorders such as acute and chronic. With that, thank you very much for your interest, and I'll be around if you have any questions.